Yeah, first of all, good evening, everybody. Just briefly, like before I start, if there are any technical problems whatsoever, may make yourself heard in some way, right? So you can give me like a a notice, in whichever way, right? And well, I mean, also, if there are questions, right, of course, we will have time for questions at the end, right? But I'm also definitely ready to take questions also in between. So it's always better to ask questions than say to shut off and say when you don't understand things anymore, right? So because all these presentations are definitely for you, right? So yeah, it's a pleasure to start this meeting. And like the one experience we had from these like online meetings is that, of course, I mean, interaction is somewhat lacking, but audiences are not lacking at all, right? So we typically have quite large groups of people and audiences, right? So as I see here now, we are 41 right now, I guess, right? And there will be maybe like say one or two more that still will join. And so it's a pleasure to present to you like some of our work. All right, so this will be a talk about polygenic adaptation. And so there is, of course, some idea why we have chosen this as an initial talk, because polygenic adaptation will be one of the focal research areas also here in Vienna in our graduate program, right? And also it's a topic that's gets a lot of attention in groups worldwide, really, right? So hope I can convince you that this is something so that is of interest these days, and also give a big background why it is of interest. All right, let's see. So before I get to any content, just briefly, who did all this work. What I'm going to present, a large part of it is from the PhD thesis from Ilse Höllinger, who has defended about a year ago in my group. And there's also some work done by Ben Wölfe, who is my current PhD student. So both in my group at the University of Vienna. And some results published in this paper by Höllinger et al. In PLOS Genetics last year. And then, so there's a second collaboration that I had with Nida Bargi and Christian Schlöderer, where we have done a review on this topic, polygenic adaptation, that came out actually this year. That's not correct. 2019, this should be 2020 in Nature Reviews Genetics. So these are my collaborators, and now let's just start, right? And let's start with this question. How does adaptation come about? And I hope you will agree that this is a question that is at the very roots of evolutionary biology. How does adaptation come about? So, what answers will you hear if you ask an evolutionary biologist? And if you do, what you will see is that it depends very much on whom you ask. There is no unique understanding about how really it happens. I mean, everybody, of course, agrees that natural selection is behind it, but this is almost already where it stops. So, if you consider the scenario where we have a quantitative trait that adapts to an external selection pressure. So here's the trait mean and here's time. And this would be, so, the change of the trait mean in time. So it increases quite a bit. The units don't matter here. We can ask how this is accomplished on the genotypic level. And then it depends whether we ask somebody who comes rather from a molecular population genetics background on the one hand, or rather from a quantitative genetics background here on the other hand. Now in molecular population genetics, adaptation, so, is usually seen as happening by what's called selective sweeps. So what's a selective sweep? That is the idea that we have adaptation or selection acting on single new mutations that usually enter the population, yeah, one after the other. And some of these are adaptive and then are picked up by selection and then dragged from a very low frequency, as you can see it here, to a high frequency. So this is time, and the y-axis would be allele frequencies. And so you always see here these rapid changes in allele frequencies from a very low starting point when selection acts on a new mutation until it then here fixes in the population. And these trajectories, all together, partly overlap, right, will then contribute to a relatively smooth change in the trait mean with time. Now, on the other hand, like in quantitative genetics, the idea is rather as follows, right? Here we have the idea that adaptation acts largely on standing genetic variation. That means alleles that are already contained in the population. In this one simulation here, they are all at an allele frequency of 0.1 initially. And then each of these many alleles has a very small effect. Then selection starts to act, and these change their frequencies, right? Mostly due to drift, actually, but positive selection makes a bias on these. So a few more go up than go down. And because these are very many, we again get a very smooth change here in the trait mean that can continue for a long time. And now you see, of course, so that this picture of sweeps here on the one hand, and of small shifts, as this is called in quantitative genetics, on the other hand, they don't match at all, right? So this is really something very different from this view that we have here over there. This is also true if we consider what's called footprints of adaptation, right? So a selective sweep, many of you may know this, generates a very clear what's called molecular footprint of selection. So this is shown here as a hard selective sweep, where selection acts on a new variant. So what you see here is, that's a stretch of DNA here on the x-axis, the y-axis. So there are just like a few aligned sequences from a population. And here we have a new beneficial mutation that has just occurred in this one individual here at this point. And the haplotype that is associated with it is drawn in red. Then, after selection, and selection will lead to an increase in frequency of this allele. It will spread in the population. Eventually, it will fix, and then you see that it drags this haplotype with it here, and we see all the population here in the vicinity of this now fixed beneficial mutation carries this one ancestral haplotype that was associated here with this original new mutation that has occurred in the population. But on the other hand, so for the small shifts picture, this would be as follows, right? So we have several variants in the population already that, on which selection then acts. And these increase a bit in frequency, certainly, as you see here, right? So these are like different loci that are visible here, right? And these increase a bit in frequency. And there are also some new mutations that also then increase in frequency. And all these together jointly do this adaptive change. But you also see here, the footprint, right, is not as clear at all, right? At each single locus, right? So here are two copies, here are 3 copies, and here is a bit of like, say, extended haplotypes that you do see in some places, but you don't see anything at all in other places. So this will give you a much weaker footprint and no clear sweep pattern. All right, so the question simply is, if we have these 2 very opposite points of view, which scenario should we expect for real traits? And I mean, as you maybe can imagine, one favorite answer that many people give to this question is, well, it depends, right? So sometimes, so it's like sweeps, and sometimes it's like shifts. We observe everything in natural populations, and to a degree, of course, this is certainly true, right? There is no doubt that we do see selective sweeps with clear footprints sometimes. And there's also no doubt that, like for many other traits, a lot of adaptation happens without leaving very clear footprints. But the question arises, what are the conditions under which we see either one thing or the other thing, right? So is this something that has to do with the number of loci? Is this something that has to do with either adaptation from new mutation or from standing genetic variation? Does the selection strength matter, whether we have weak or strong selection, or the strength of the mutation rate also? So, which scenario is favored under which conditions? You can think this for yourself, right? So which condition do you think, right, would be the most important one of these? And we will come to some clues in the end of this talk. All right, so I'm a theoretician, and theoreticians need models, right? So, and therefore, I'm now going to walk you through the model that we've used here for this work, which actually is an absolute standard model of population genetics, right? So we assume we have what's called an additive quantitative trait under stabilizing selection. So this is the trait mean, right? So these are allele frequencies pi at L loci that contribute to the trait, and the locus effect, we assume here, is equal effects at all loci is gamma. This is the fitness function that acts on this trait, which is just a Gaussian fitness function for stabilizing selection. That has a shape, that looks like this. We look at n haploids to keep it simple. We have L biallelic loci here, and we have a recurrent mutation rate at all these loci. That is given by these population mutation parameters. All right. So initially, this is the trait axis, and here's fitness, and we have an optimum at some value z0 before the environment changes at some point, right? And then what I said, right? So there is a sudden environmental change, and this optimum changes from this old position here to a new position. So this happens suddenly, and then adaptation, of course, will drive the trait mean from this old position here towards this new optimum. And it will reach this optimum at some point after maybe quite a long time. But what we do is, so we will record just at some point where the optimum here is right now at the value of Z1. There we will assume that we sample this population, right? And we will just see how this adaptive change from here to there has been accomplished on the genotypic level. So at this position, what we will do to assess how this has been done, we will consider what's called the joint distribution of allele frequencies pi here at this locus, this pi at this point, And this way we see which loci have contributed, and we will do this over very many replicates, and this way get an idea of, for example, whether typically only few loci contribute or whether many loci contribute. And this is also what we call the architecture of polygenic adaptation. All right. Now here are a few slides of how we approached this mathematically. Won't be too complicated, and also like if you get lost here, right, I'm going to pick you up again afterwards, so no worries, right. This is just like to give people with a bit more of a theoretical mind an idea how we have approached this, right. Now this is the idea. Like initially, at least like if adaptive alleles enter population by new mutation, they are at a very low frequency, right? Or let's assume also in the standing genetic variation they are at a low frequency if they are initially not neutral, but maybe slightly deleterious, right? So they are then at a low frequency. And initially, so what selection does, it picks these guys up, drags them to a high frequency, and then like once we approach the new optimum, right, so then all of a sudden this change gets slower, and so I mean this is frequency here, say on the y-axis, and these frequency changes get slower, and at some point they are altogether stopped, right? So and then we can dissect these phase of rapid phenotypic adaptation again in two parts. As I said initially, these allele frequencies are really small. We call this the establishment phase. And when allele frequencies are small, then the movement of these alleles is mostly stochastic, right? It's mostly determined by mutation and drift. So these are the most important processes initially. And so we'll set up a theory that is just governed by these two forces. Later on, when these frequencies are larger, then drift is no longer so important. Selection then is very efficient. The movement of these alleles becomes deterministic. And so we can describe this by a deterministic theory, where selection really, and also epistasis, the interaction between loci plays an important role. Technically, so this is a branching process, and this is a differential equation later on. So this initial phase with a branching process, say, just to give you a short idea of that, right? What we use here is a so-called Ewell branching process, where we track single mutant copies that are destined for establishment. So we only, like when a new mutant copy arises, we initially determine whether this will get lost again due to drift, or whether selection will overcome drift and it will get established. And we focus only on those that do get established. They get established with a probability PS, the establishment probability, right? And then the things that can happen is, in this process, forward in time, is that new lines in this branching process arise, and this will always happen by new mutation, right? So new mutation happens, and also we need to decide at each new mutation whether it establishes. So we have a product of mutation rate and establishment probability for the generation of new lines. And then once we have these lines, of course, they can also split, generate more than one offspring, right? That's called reproduction. And with each reproduction event, right, reproduction occurs here at a rate of 1, we again need to ask whether the newly reproduced individual will establish, and only in this case we'll include it into our theory here. So therefore, the split rate in these U trees is proportional to also to the establishment probability. All right. And okay, so we see this mutation and yeah, these reproduction events during establishment, they create stochastic differences in the number of lineages that we see at various loci, right? So this is a first locus here, this is a second locus, and in the end we want to count the number of lineages that we have at each locus, because this tells us something about the allele frequencies. But the one interesting thing is that these ratios here in this number of lineages, right, they always depend on the mutation and rift here, but they are independent of the selection strength. The reason is that here this selection strength is something that enters these establishment probabilities, but all events that happen here in this small process are just proportional to the establishment probability. So whether this is large or small then just matters for the time scale on which things happen, but not on the relative success at different loci. All right, and this way we are able to derive some mathematical formula that gives us a joint distribution of frequency ratios, right, at all these loci. And this is something that is independent of selection, but only depends on the mutation rates. Second phase then is like deterministic dynamics. And this deterministic dynamics is one that describes us the approach of the new optimum that is under stabilizing selection, right? And this has like the equation here has like two parts of it. One that describes here directional selection. So if this is here, so if the selection optimum is still larger than the current trait mean, right? So then this term is positive, and this will lead to an increase in the allele frequency at like all these loci, right? An increase in the allele frequency that drives the phenotype in the correct direction towards the optimum, right? So this is a term of directional selection. On the other hand, there's a second term, and this is disruptive selection on the level of single loci. And this may be a bit surprising at first to think why does stabilizing selection on the level of the trait lead to disruptive selection on the level of single loci. Now the reason is, once we are really very close to the optimum, right? Then alleles, if they adapt, can overshoot this optimum, and then mainly what happens is that there is selection against genetic variation, right? And how is this done? Selection against genetic variation. This is done by moving alleles that are already at a high frequency up to frequency 1 and moving alleles that are at a very low frequency down to frequency 0. So this is disruptive selection on the level of single loci. However, you also see that this term here only matters if this distance here of the optimum to the trait mean is something that is of the order of this gamma here, right? So we have one gamma here, we have a gamma square here. So if this parenthesis is something that is larger, much larger than this gamma, so this, which means that with the distance to the from the trait mean to the optimum is still much larger than the single locus effect, then we can usually disregard this second term. And this is what we will assume from now on, right? And then it says we have a model of directional selection. And with this model, we can do like another, like say, parameter transformation. And I won't go into like great details here, but only like, yeah, say, to tell you that now again the relative change in these transformed allele frequencies at different loci then is again 0. So this is something that then tells us, once we know these values initially, right, so we also know the outcome. All right, so maybe this is an equation, right, so that has been derived from this model of stabilizing selection, but actually our theory holds for a much larger group of models where we can have any kind of direction selection term here. So replace this term here by something much more general, and the same results will still hold. All right, a nasty formula comes out of it, right? And the only thing that I want to point at with this formula is that, again, this is something that is independent of locus effects or selection strengths. So if you look closely, there are no gammas or no sigma in this formula, but it only depends on these thetas. So here are thetas, everywhere are thetas, right? And so these are these mutation parameters. All right, just keep this in mind. But now immediately we leave the maths and go to the results again. All right, so let me summarize assumptions now that I make for the results slides. We assume that we have loci that are not only equal strength, but also have an equal mutation rate in all these loci. It's not an assumption we need to make, but we just make it for simplicity for this talk. We also assume that we start selecting from mutation selection drift balance. So that is a generic assumption for an initial value, right? And so we have an initial trait optimum at Z0, and say we follow this adaptation from standard genetic variation and also new mutations that are allowed until the mean phenotype reaches a new threshold value at Z1, as I've shown you initially on the model slide. And then, say we look at all the loci that do contribute to this adaptive change, and we have these loci always ordered according to their contribution to the adaptive response. The first locus is the one that contributes most, the second locus is the one that contributes second most, and so on, right? And the question is, what kind of architectures can we observe? Now, let's consider one scenario here, right? And so this is a scenario where we have a trait that is made up out of 10 loci. And we assume that we change the optimum from an initial optimum of 0 to a new optimum that is 3.333 in units of a single locus effect. So this is what this means, right? So, all right, so we change the optimum from 0 to 3.333 locus effects. So how can we accomplish something like that? All right, so this is an obvious way how we can accomplish this, right? So what you see here is, right, on this axis here, this is allele frequency on the x-axis. And on the y-axis here, this is the density of the frequency at the various loci across many replicates. So what this tells us here is the following, right? So we see here a spike here on this side. And actually, right, so different loci are different colors, right? And if we look closely, that there are 3 loci, 3 colors on top of each other here, which basically means that 3 loci have walked all the way from frequency 0 to frequency 1, right? So they have they've reached fixation in the population. And then there's a third locus, so that has that has also started here, right? And has now walked to just 1/3 here in this frequency in this frequency range, right? And so these three together plus this one just make this 3.333, right? So that we need for our overall adaptation of this trait. And then there are 6 other loci that didn't even have any variation, right, to start out with. So this is one scenario, one possibility. And this is something that we would call single sweeps, right? We have 3 sweeps that are completed, and we have one that is still a partial sweep, right? So this is, it's on its way. If we would wait for longer, then it would move over here, right? But right now, when we've sampled it, so it's still a partial sweep. Oops, other direction. All right, but sometimes we also observe something like this. So still we have 3 guys that do most of the work, right? And so there's a third one that still here lingers around, usually, right, in the neighborhood of a frequency 1/3. But you also see this color stretches out in this direction and also this direction. So sometimes this third locus is also much higher frequency, sometimes at a lower frequency. And then there are here that also contribute here, like a 4th and a 5th color, right? Which are still other loci that also contribute maybe a little bit to this total adaptive change. So up to 5 loci here usually contribute for these parameter values. So in this case, then this means we have single sweeps plus some scatter, we can maybe say, right? But still it looks relatively sweep-like. Let's just continue this, right? Now here we have a pattern where even more loci contribute, right? And all these things has now smeared out a lot more, right? We see a lot going on at these intermediate frequencies, but still there are also some loci that have at least almost reached fixation. So one could call this completed sweeps, some here, but also a lot of partial sweeps, so that dominate this adaptive pattern. And then if we go on, right, now we see something, right, that all 10 loci now contribute, right? And we can still clearly say, one locus contributes most, as a 2nd, 3rd, 4th, and so on, right? But they cover a whole range of allele frequencies, right? And all contribute to this adaptation. Finally, right, there's a situation, something like that, where there is no single locus anymore that would make a sweep or reach fixation here, right? All are stuck at intermediate frequency values. And also, right, so these are distributions that are widely overlapping. And so something like this, we would call them collective polygenic shifts, right? So this is the pattern that's predicted rather by quantitative genetics, whereas of course the sweep pattern here, right? So this is the molecular population genetics pattern of adaptation, right? So we have it all here in our model, right? All these different patterns. And now, of course, the question is, what are the important parameters? What makes this difference, right, from one pattern to the other? And the answer is that this is a single parameter. A single parameter determines the adaptive architecture. So whether it looks like this, or like this, or like this, like this, or like this here, right, is determined only by this parameter theta background. It's also called the background mutation rate. So what is the background mutation rate? It's just like the per locus mutation parameter times L minus 1, where L is the number of loci that contribute to the trait. And so we have here L minus 1. Why L minus 1? Now, well, if we look from the point of view of each single allele that happens, each single adaptive allele that happens at a certain locus, right? Then this single adaptive allele wants to run its course to full fixation maybe, but it gets competition, right, from alleles that occur at other loci. And so there are L minus 1 other loci where alleles can compete with our focal allele. So theta background measures competition that every sweep locus receives from alternative redundant adaptive alleles in the genetic background. Right? And here on this side, the theta background is really small. So this should, of course, be 0.01. Yeah. So this means if a new adaptive allele occurs at one locus, it very rarely gets competition from these other loci, and therefore it just proceeds to fixation. But then, once this theta background increases to 0.1, 1, 10, and 100, right, we get quite a bit, and then really a lot of competition from these other loci, right? So, and then alleles start to like increase in frequency at very, very many loci in parallel, right? And then, of course, like, so we get a collective picture where very many loci contribute to the adaptive response. So, and each single one does only a small shift. So, sweeps on this side could call this partial sweeps here, and we have shifts here on that side. All right, a bit more to the adaptive architecture, what we find here. What I'm now, like say, going to show is how this looks like for the additive trait and a single adaptive step, right? So here we have 4 loci, here we have 10 loci, and here we have now 100 loci. Remember, right? Before we had all these pictures were for 10 loci. Now we include also a trait with few loci and a trait with many more loci. We'll always assume, like that adaptation does only a single adaptive step. Here it goes from an initial optimum of 2 to 3, here from 5 to 6, here from 50 to 51. We'll consider this for 3 different values of the background mutation rate, much smaller than 1, 1, and much larger than 1. And so this is what we get and what we see. We see that theta background at 0.1, it's this sweep-like pattern, right? So where one locus does this change almost alone on this side, and all the others sit here. So there's nothing really in the middle. And this is true whether we have 4 loci, or 10 loci, or 100 loci. So the number of loci doesn't matter. Once we keep this theta background fixed, the number of loci doesn't really change the qualitative picture of the genetic architecture. On the other hand, for theta background at 10, we always have a lot in the middle, like here, or here, or here, right? And so this is more like a shifts-like pattern, right? It gets even more shifty, right, if we go for theta background to 100, right? But the main point here is that we have always a lot of loci, right, that collectively contribute to the adaptation and only have smaller allele frequency changes. And then, of course, like at theta background equals 1, we have something sort of intermediate, which I usually call like partial sweeps. So this, of course, was now only like a single adaptive step, like going from like 2 to 3, 5 to 6, or 51, or 50 to 51. Right? Of course, we're interested in the case where we now do adaptation over larger distances. We shift this trait optimum, like say, over large distance. Now again, we focus again on this case of 10 loci because I can tell you for a different number of loci we get equivalent results. And we'll change really like say the optimum from one end from 0 to 10, right? We're covering the whole range, right? So this is the change in the optimum. But we will look at the genetic architecture once the trait mean has reached certain threshold values at 1, at 3, at 6, or at 9, right? At these different values, right? And then this is what we see. And this is now like a picture that I have to explain you in like a little bit more detail. Maybe we will focus first here on this first row. This is like easiest. Again, what you see is stuff at the left and stuff at the right, always, and nothing much in the middle. I mean, so these dots I will just explain to you, but there are no allele frequencies here somewhere in the middle. And this means we apparently have just selective sweeps, right? And now these colors and dots. So this means, so these are the 10 loci that we have here. And the ones that are colored are always like the allele frequencies that are displayed here in this figure, right? So red, this has almost sweet fixation. So this is a locus that has swept. This is the largest locus with the largest allele frequency. And blue, this is the locus with the second largest allele frequency. This is still here at frequency 0. And of course, all the others, right, have even smaller allele frequencies, right, and therefore, like, are just crumbled together here, right, somewhere at around frequency 0. Here, right, all the black dots, these are ones, I mean, all these, like the colors, the blue, these are the ones that are displayed. One is again almost a fixation, and the other one here is at a very, very low frequency. And here 2 black ones that sit on this side and have also like already fixed, and all these white ones sit on this side and have not even started to run, right? And similar here and similar here. So you see, right, it is just one locus after the other flips from frequency 0 to frequency 1. Right, so here we need 1, and 1 does the job. Here we need 3, and 3 do the job. Here we need 6, and 6 do the job. And here we need 9, and it's 9 who do the job. So one after the other just contribute to this adaptive change. And all this is for theta background 0.01. Right. Now for theta background 0.1, and for 1, we then see, right, that of course now we get more of these allele frequencies in the middle, and therefore we have adaptive architectures. So that do not only have sweeps, but we also see that if we move from this side to this opposite side, right, here we still have quite a lot in the middle, but then here and here and here it gets less and less, and this is almost a sweep-like pattern in the end. So something changes here a bit. Also here, we have a lot in the middle, like say initially, and then this gets a bit reduced and even more reduced. And here, so again, maybe this is a bit similar like this, but this is almost like a sweeps or partial sweeps scenario that we would see here. So question is, how does this come about? And the answer is that, I mean, here, for the first one to move from this side to this side, right, this one sort of has 9 others to compete, right? So this theta background is theta times 10 minus 1, 9 times theta, right? But like if we are the 6th already, right, who need to do the next step, right, so then say 5 are already here on this side anyway, so they don't compete for the next step anymore, right? I only have like 4 others, right, so that would compete. So my effective theta background, which counts the number of competitors, is smaller, right? And therefore this gets reduced from one side to the other. And therefore parents, as we walk along, and increase this trait mean ever more, right? We sort of run out of adaptive material slowly, and then patterns that are initially more shift-like get more and more sweep-like. So this is what we see. All right. And now already, let me summarize what we've seen. So I mean, we did some mathy stuff, and we found analytical approximations for the joint distribution of mutant frequencies, yeah, and assuming haploglossy with equal effects, and also assuming, yeah, I didn't point this out, so much linkage equilibrium among these loci, right? However, the locus mutation rates can also differ, and we get analogous results. We can treat different trait architectures, so we don't necessarily need to have this stabilizing selection with a shift in the optimum. We can describe almost any variant of directional selection, initial directional selection, right? And we can also extend the theory to diploids and also to include linkage, right? So this assumption of linkage, this equilibrium really is not an important one. In all these cases, we see that the adaptive architecture is independent of the selection strength of parameters sigma for the strength of selection, parameter gamma, which is the locus effect, also the value of this trait optimum, but only depends on the mutation rate, right? So this theta background parameter is really the master parameter of this model. Then, of course, there are some open questions, right? As I said, so far we assume equal locus effects. If there are unequal locus effects, then these results should really change a bit. And also, of course, if we have population structure, if we have local adaptation, more complicated adaptive scenarios, right? But there's always more work to be done, and I hope that, for example, I can do some more on it with Ben, for example, who's my current student on this project. So, if we believe for once that this background mutation rate, right, is the important parameter here, we can now classify several regions where we find qualitatively different genetic architectures. And this is like, so if this theta background is something that is smaller, like something like, say 0.1 or so, then typically we get selective sweeps, like predicted in molecular population genetics. And so traits can still adapt over large distances, then we need a lot of sweeps, but they will occur as clearly visible sweeps with also clear footprints. Then on the other hand, if this theta background is large, larger than something like 10-ish or so, right, then we get here these small frequency shifts at many loci, no clear selection footprint in the variation. And then in an intermediate range between 0.1 and 10, and then we have like say heterogeneous pattern of polygenic adaptation with a lot of partial sweeps, some completed sweeps, and like a lot of loci that still linger around at intermediate frequencies. And this is like a mode that like is not so well described so far, right? And it covers an interesting biological parameter range. And therefore, also this is a task to really describe this intermediate range of background mutation rates. All right. So one question that arises in applying this theory to real data is how should we explain adaptations by small shifts that we seem to see for a lot of traits that people in quantitative genetics like to look at. For example, traits, body size, weight, or yield, that are highly, highly quantitative traits, right? And so apparently we need for all these cases, quite large values for this theta background, right? And now it turns out, of course, that here this theta background has several factors in it, right? So there's this L-1, there's this NE, and there's this U. Right? And each of these factors could in principle be the one that is responsible for the whole thing being large. Right? And one thing definitely is if L is super large. If this is larger, something like 10,000, right? Then we have like a large omnigenic basis, as this is called, for quantitative trait. And then this combination here can easily be large because U, right, the mutation rate, this is something that's typically super small, right? And even if like NE is large, NE times U is also typically something that is still quite a bit smaller than 1. So we need a large genetic basis to get the whole thing really larger than 1. An alternative is that really this value NE is much larger during the time when we observe the adaptation than the NE that we typically estimate from within population diversity. And this can also be true, right? Because within population diversity, as you may know, is affected also by ancient bottlenecks. Whereas, right, the adaptation that happens here and now really, say, only feeds the population size that we have here now, which may be larger than this effective population that we often work with in population genetics. All right, so this is sort of a within model explanation, right? So we just say in this model that V is the master parameter. This parameter here in our model is really large. But of course, it could also be that our model is just too simple, and there are two possibilities that stand out here as alternative explanations. It's that these initial allele frequencies are really much more homogeneous than predicted by mutation selection drift balance. I mean, remember, this is one assumption that we had, right? So that we start out with mutation selection drift balance. And then, we have an initial heterogeneity, like among allele frequencies at different loci. And therefore, also, right, some of the loci will contribute a lot and some not so much. But if we have this super homogeneous among these loci, then it's easier that more loci contribute all a little bit, right? And then it goes more in the direction of small shifts at many loci. And this could be due to some reasons like balancing selection initially, or some spatial structure or a mixture that we have. All right, but then another important aspect is that definitely, so we have more than just these extreme examples of adaptation by sweeps or adaptation by shifts on the other hand, right? So these are just really only the extreme endpoints on a continuous scale of patterns that can really happen. And there, what we really want to do is to describe these heterogeneous patterns of polygenic adaptation much better. These regions are partial sweeps predicted for a large parameter region, and so we expect that we get very heterogeneous patterns among loci. Some contribute a lot and some not so much, right? And also among replicates. So if we do this in experimental evolution in many replicates, we also assume that different replicates really look differently. Can I ask, is there any evidence? Yeah, well, of course, there's a lot of evidence for partial sweeps, definitely, right? It's even like that completed sweeps, right? If you look at the data, right, really completed sweeps are rare in natural populations. And we often see a plateauing of allele trajectories in experimental evolution. So something like this partial sweeps, that get stuck at these intermediate values, what you see here for these bluish guys, right? So is something that we do see in data. And also, we definitely see that the response is strongly heterogeneous among replicates. So if we do this in different replicates and repeat the selection experiment, different loci typically react, as we have seen in Niederbagi's paper in PLOS biology from last year. All right, so this is what I had, right? So my thanks goes to these people, of course. Funding for these things come from our PopGen Vienna Graduate School. And thanks to you also. And now we still have a little time for questions.